مرحبا، اليوم رح نحكي عن مشروع مادة الموديلينج اللي هو بعنوان Modeling of Kidney Hemodynamics.
مرحبا، أنا الطالب كريم خصاونة، وهسه رح نبلش نحكي عن مشروعنا.
طبعا هسه أول شي رح نبلش في الانترودكشن. The most important feature of the vascular system is that it's highly branched. It is assumed that the branching geometry of the blood vessels is conferred by defined principles. Tree-like structure of arterial system are subjected for experimental and theoretical studies. Structural connectivity in vascular networks directly influences their functional state. The network experiences local responses affecting local blood flow and altering the overall state of network. The topology of the vascular network is also an important determinant of the consequences of pathological processes in the vessels. The nephrons are arranged in a complex branching structure. The network delivers blood to each nephron and it provides spaces for an interaction between each nephron and the neighboring ones. The interaction between is a complex spatial patterns of synchronization between tubular pressure oscillations of the individual nephrons. Nowadays, both functional significance and spatial patterns and the mechanisms leading of their formation are unknown. An important step in the investigation of the mechanisms and the functional significance of spatial synchronization of cortical blood flow is better understanding of topology and renal circulation. This requires a detailed mapping of the entire renal vascular tree. Features of nephron vascular network are: the first one is a significant pressure drop between the origin afferent arteriole and its insertion into glomerulus. The second feature is the pressure of the tubular glomerular feedback mechanism. By experiment, neighboring nephrons interact by vascular propagation of electrical signal initiated by TGF each nephron, which travel via endothelial signal cells in the arterial wall and can reach the neighboring afferent arteriole. The coupling tends to produce simultaneous constrictions of the afferent arterioles, leading in phase synchronization of the oscillations. And now we are going to talk about the models. Several recent model studies have investigated features of nephron-nephron interactions. The complex renal arterial network in these studies was approximated by a simple bifurcating tree-like structure and included a group 10 to 20 nephrons. And these are the graphs for the network topology. The left graph is the relation between the daughter vessel diameter and the parent vessel diameter. The graph on the right is talking about the relation between the vessel diameter and the vessel length. We assume that the distances between the origins of AAs and the vascular tree can be described by an exponential distribution. This assumption is based on several observations. AAs often originate in doublets or triplets, even from large vessels. This implies a high probability of zero or close to zero distances between neighboring AAs. Vessel segments without AAs branching from them are rarely seen. This implies a low probability for distances between AAs of several hundreds of microns. The simplest possible distribution that satisfies observations 1 and 2 is an exponential distribution. Finally, we assume that the distribution of the distance between neighboring AAs is independent of the diameter of the feeding vessel. And here we obtained two 3D stacks of renal vascular structure from which we measured the distances between the origins of neighboring AAs. Hemodynamic interaction. Hemodynamic interaction occurs when one nephron is stimulated by TGF mechanism to contract its afferent arteriole, causing the hydrostatic pressure to rise at the upstream branch node, increasing the blood flow to the second nephron. To describe the flow in each vessel of the vascular tree, one needs the hemodynamic resistant RHTR calculated from Poiseuille relationship.
مرحبا، أنا الطالب شيماء جنايدة. طبعا هلا رح نحكي عن ثاني موديل بصير عنا اللي هو الإلكتريكال انتراكشن. فالإلكتريكال انتراكشن equivalent electrical circuit a vessel segment light gray located between two branch points. The activation in TGF in a nephron causes a membrane depolarization and a contraction of the smooth muscle corresponding to the afferent arteriole. The cell of the renal vessel are coupled electrically by gap junction. Gap junction هو المربع اللي باللون الرمادي بيمثل GG, GC and CC. The cell of the renal vessel constitute a well coupled with the endothelial cell form the path least resistance. IN stands for the current induced by to the macula dehiscence signal. V previous بتمثل described to the voltage into the previous downstream. CU is the unit of the capacitance. GU and GG are the unit of the gap junction constant use respectively. هلا رح نتعرف على differential equation اللي رح يصيروا بهذا الموديل. في عنا 3 differential equations. The first one for segment sides that are connected to the nephron. V previous described to the voltage into the previous downstream. V rest is the resting membrane potential. The next one for the inner units of the segment فيها شغلة بدنا نوضحها اللي هي V next donated potentials upstream units. IN stands for to the current induced by to the macula dehiscence signal. بالنسبة لثالث معادلة اللي هي at the branch point downstream of the vessel فيها الCU رح تمثل عنا الكاباسيتانس. الGU and GG are the unit and the gap junction coincidence respectively. هلا رح نحكي عن single nephron model. فهون في عنا بوضح بالشكل التالي. في عنا أول نقطة اللي هي proximal tubule PT رح تكون بعيدة عن الجيوميرس بينما الديستال تيوب رح تكون قريبة منها. أيضا في عنا أسهم بتوضح اتجاه حركة الفلو من X1 to the loop of the henial إلى X2 بعدين بتروح للبادي. في عنا مجموعة من differential equations single nephron differential equations موضحين عنا. أول واحدة منهم the first equation of the model فيها بعض المتغيرات مثل الأفلت is the single nephron glomerulus filtration rate. Proximal tube FREB is assumed to be constant. Flow into the loop of the henial is determined by the different PT ناقص PD. C tube is represented to the elastic compliance of the tube. Flow resistance R panel. The second equation represented and described the dynamic of the afferent arteriole. R small represented the inner radius of the vessel and V small is its rate of change. Sigma is a measure of the mass relative into the elastic compliance of the arteriole wall. BEQ is the value of this pressure for which arteriole is in equilibrium. BAV and BAQ involve a number of algebraic equations that must be solved. The last differential equation describes the delay T and TGF regulation. In the single nephron model describe the delay T and TGF regulation. This delay arises both from the tensed time through the loop of the henial and from the cascade enzymatic process between to the macula dehiscence cells and the smooth muscle cells that control the contraction of the afferent arteriole. رح نشوف هلا example بيوضح differences بين ال ABT وال KSABT. ففي عنا بعض statistical data بتوضح هاي differences بالafferent arteriole. The larger diameter of the feeding vessel in the KSABT structure reduce the thermodynamic resistance of the vessel and consequently the pressure drop from the renial artery to the afferent arterioles will be smaller when compared to the ABT structure. Another conversation between to the ABT and to the KSABT structure. فالcomposition of the net flow in the afferent arteriole and to the glomerulus filtration. فزي ما هو واضح برسمة ال ABT بيطلع قاعد بزيد بشكل منتظم وبيطلع للأعلى، بينما برسمة ال KSABT بيزيد بعدين بيوقف لحد معين بعدين بيرجع بيزيد إلها. Discussion. The systemic circulatory system plays a central role in supplying organs and tissue with oxygen and nitrogen and removing multiple waste products. In the kidney, the circulatory aspect of the circulation is not only to supply the tissue with oxygen and nitrogen, but rather to support the filtration of the plasma at the glomerulus of each nephron. Imaging modality, MUCT and conventional microscopy have made it possible to obtain images to then either microcirculation in a given origins. In this study, we present new proximal finding of the 3D arteriole and the afferent arteriole structure, and we developed a probability-based algorithm for the generating such structure from the data. Expositional distribution are typically seen in Bayesian process where the time of the distance between events are independent for each other. The microcirculation of the kidney is usual because it has two capillary network and the glomerulus capillaries and the vertebral capillaries connected through the afferent arteriole. هذا الشكل بيوضح ال effects of the electronic interactions. في عنا بالشكل الأول بيوضح التداخل والاختلافات بشكل كبير بينهم، بينما بالشكل الثاني المسافات بينهم أصغر.
بسم الله الرحمن الرحيم. أنا الطالبة أثير أحمد الخطيب. Another important feature is that asymmetrical connections of endothelial cells can lead to anisotropic propagation of electrical signal between two afferent arterioles and can affect nephron-to-nephron interaction. This figure shows the simulation results for ABT in the left side and the KSABT in the right side for initial diameter equal 40 micrometer and stop diameter equal 22 micrometer. Another possibility is that the individual vessels actively adjust their radius to compensate for differences in the feeding pressures. This mechanism plays an important role in renal autoregulation of blood flow and is implemented in our model in a simplified way. An anatomical feature not explicitly included in the present algorithm is the presence of the triplets and quadruplets at the branch site. About the stability, to test the stability of the model, we ran simulations for different values of parameters such vessels compliance, beta and alpha. To choose materials and methods, an experiment, a 12-week-old Sprague-Dawley rat was anesthetized. A catheter was inserted into the aorta and the vena cava was opened. By the way, the Sprague-Dawley is a kind of the rats used in virtually all disciplines of biomedical research, including toxicology and pharmacology. About the computational implementation, this algorithm combines a recursively bifurcating algorithm and recursively bifurcating algorithm with additional segmentation. And this figure shows the functional diagram of the algorithm. Simple recursively bifurcating algorithm. First, calculate the length of the first vessel using initial diameter and GVLVD distribution. Second, calculate the diameter of the first datar vessels using GDDP. Finally, calculate the second datar diameter using the parent diameter The first that are diameter and Murray's law. This description of electrical coupling represents the main notations of the coupling. Each endothelial cell is described by the wall cell conductance GC, which can be linear or nonlinear and instantaneous or inertial. This figure shows the schematic representation of electrical coupling. This description of nephron model, the first equation to determine the flow in the loop of Henle, the second equations to determine the preglomerular resistance. A, B, C, and D are found from the model equations, and we solve the third order equations to find CE. And we use these equations to determine the functions of a plasma protein concentrations. And we use these equations to determine the tubular-glomerular feedback function. Finally, we use these equations to determine the equilibrium pressure. But notice that the perimeter PA in the single nephron model becomes a variable in the nephron arterial network. Thus, we have reached the end of the project. Thank you.